Good day and welcome back to the Phlying Doctor Channel. On today's video, we're delving into a scientific study that looks at the Zika virus and an enigmatic relationship between that virus and some of the cellular processes that occur within each and every one of our cells. This study provides more evidence to the growing field of virology that Zika virus changes gene expression, but also exerts a profound effect on a process known as alternative splicing. As a whole, this study introduces the concept that Zika virus-induced alternative splicing may be behind some of the more devastating neurological complications associated with the most recent Zika virus outbreak. So, sit back, relax, get ready to learn. Before we actually get into today's study, we first need to set the stage. I'll first introduce Zika virus. We'll take a trip down memory lane and find out where and when the virus was actually discovered before we introduce some of the more common symptoms associated with infection, and then we can get into the meat of today's study.
Zika virus is a mosquito-borne flavivirus that gained global attention in the 2010s due to the severe neurological complications it caused, including microcephaly and Guillain-Barré syndrome. The story begins several decades earlier, however, on the African continent. In the 1940s, the Rockefeller Foundation was supporting investigations into the ecology of yellow fever. As part of this research, scientists would utilize sentinel animals strategically placed amongst different geographies. On weekly or semi-weekly intervals, samples from the animals would be taken and screened for yellow fever or any other disease that the scientists were studying. If during the sampling an animal was found to be ill, the animal was brought back to the main lab to be properly cared for and studied. Researchers working for the Rockefeller Foundation set up multiple rhesus monkey sentinel animals within the dense jungles of the African Zika forest. In 1947, monkey number 766 was found to be feverish, and serum collected from this monkey did not neutralize the virus these researchers were studying, yellow fever. In fact, it appeared this was a previously unrecorded virus.
As was tradition, the virus was dubbed Zika virus, in reference to the geographical region where the virus itself was isolated from.
The strain of this particular virus was MR766, in reference to the rhesus sentinel animal number 766 the virus was isolated from.
For the first 6 decades, the virus remained relatively hidden, with only a handful of human infections documented. This era of silent transmission could be in part due to a number of reasons. There is a lack of reportable infectious diseases data from the region where this virus was endemic and isolated from. Second, most people who become infected with Zika virus don't actually show any symptoms. In fact, for the 20 to 30% of people who actually show symptoms and are infected with Zika virus, it's often very mild. Many of the symptoms overlap with a number of different mosquito-borne diseases that circulate within Zika endemic regions. This complicates the reporting aspect of controlling infectious diseases.
The first recorded Zika virus epidemic occurred on the isolated islands of Yap, part of the Federated States of Micronesia in the Western Pacific. In April and May of 2007, an outbreak of dengue-like illness caught the attention of local physicians due to its atypical clinical presentation in some patients. Subsequent testing confirmed Zika infection. This outbreak affected approximately 75% of the population, with all reported cases being mild.
Following the outbreak on Yap, there were no reported Zika virus outbreaks until 2013. At this time, cases began emerging within French Polynesia. Later that year and into 2014, the situation escalated into a major epidemic, totaling over 30,000 cases. The virus then spread to New Caledonia, the Cook Islands, and Easter Island, and throughout the South Pacific region. In November of 2015, a major outbreak of neurological complications in newborns was discovered in Brazil. This was later attributed to Zika virus. From Brazil, transmission of Zika spread north throughout Central America, the Caribbean, Puerto Rico, and the southern United States. So, now that you have a better understanding of the origin of Zika and the contrasting symptom severity associated between the original Zika virus isolated from Africa and the more modern Zika virus in the Americas, we can dive into today's study and discover what the scientists found. The study we are discussing today aimed to understand how Zika virus interacts with our cells on a molecular level.
The scientists asked, compared to a similar flavivirus or the first isolated Zika virus, does infection with a modern Zika isolate cause changes within cells that may explain some of the more devastating symptoms seen recently?
To do this, researchers used a neuroblastoma cell line called SHSY5Y and infected them with two different isolates of Zika virus. One strain was the modern Zika virus from Puerto Rico, associated in 2015 with the current outbreak. And the other was the original Ugandan strain, isolated back in 1947, named MR766.
To ensure the results the scientists found were specific to Zika virus and not just a viral infection, the scientists also used a similar flavivirus, dengue virus 2. This control would allow the team of researchers to determine which set of genes were differentially expressed due to viral infection or were specific to Zika infection.
The scientists first confirmed that the SHSY5Y cells were susceptible to each of the three viruses. As you can see in these images here, each individual foci of blue that you see is an individual cell. If we overlap this green-colored double-stranded RNA, which is an intermediate in the viral life cycle, you can see that the distribution of green, or the amount of cells that are actually infected with virus within each of these images, is relatively similar. Approximately 31 to 40% of cells between the three different viruses were infected, suggesting that there was no bias between the viruses in terms of infectivity. These particular sets of experiments were essential in justifying any downstream conclusions drawn from experiments.
Even though the infectivity and RNA levels isolated from infected cells were similar between all three infections, the team of scientists
Asked whether cells infected with the different viruses shared similar patterns of gene expression using RNA-seq. They collected data from the three sets of experiments and obtained millions of reads, which they mapped to the human genome. Using specialized software, they quantified the gene expression between the different infections. This revealed a significant difference in the transcriptome when infected with one of the viruses. The results were striking, with 1,464 genes differentially expressed when cells were infected with the modern Zika strain compared to uninfected cells. This was far more genes differentially expressed than when cells were infected with the original MR766 Zika virus strain or dengue virus 2. Looking more closely at which genes were actually differentially expressed, they used a process called gene ontology analysis. Gene ontology analysis allows researchers to categorize different types of proteins or genes into functional groups. When the scientists performed this gene ontology analysis, they realized that the modern isolate of Zika significantly changed the expression level of immune response genes. When comparing the top 10 differentially expressed genes between each of the three different infections, they noticed that all of the genes that were differentially expressed within the modern Zika isolate were all upregulated. When they looked at the original Zika isolate and the dengue virus 2 isolate, they noticed that genes were mixed in the top 10 category between up and down regulated.
These results affirmed the researchers' hypotheses and indicated that the more modern Zika viruses circulating resulted in much more significant changes at the molecular level.
The scientists looked at 5 major categories of alternative splicing. Problems in alternative splicing can lead to diseases when sequences necessary for proper splicing are altered or when regulatory factors essential for splicing are mutated. These issues can result in missplicing, leading to reduced functional protein production, or an imbalance in mature mRNA isoforms that contribute to disease. Before we dive into the actual results as they relate to this study, let's take a closer look and figure out what exactly alternative splicing is.
To understand alternative splicing, we must start at the beginning, in the nucleus of our cells, where DNA encodes the genetic blueprint of life. This DNA contains genes which serve as instructions for building proteins, the workhorses of our bodies. Genes are made up of exons, the coding regions, and introns, the non-coding regions. When our cells need a particular protein, a process called transcription occurs. An enzyme called RNA polymerase reads the gene's DNA code and creates a complementary RNA molecule known as pre-mRNA. Here's where the magic happens. The pre-mRNA is full of exons and introns. Alternative splicing is like a skilled editor deciding which parts of the pre-mRNA should be included in the final mRNA transcript. What's fascinating is that alternative splicing can result in multiple mRNA transcripts from a single gene, leading to the creation of diverse proteins with unique functions. After the mRNA is edited through alternative splicing, it is exported from the nucleus and goes to the ribosome for translation. Here, amino acids are brought in and our unique protein is synthesized. So, there you have it, a very brief introduction to alternative splicing.
It's a remarkable process that adds a layer of complexity and versatility to the way our cells operate and makes you who you are. After compiling the RNA sequencing data, the researchers came up with about 77,000 potential unique alternative splicing events. They found that the more modern Zika isolate resulted in at least 2.5 times more alternative splicing events than the original Zika from Uganda or dengue virus 2. These results suggested that there was a substantial imbalance in the splicing landscape when cells were infected with the modern Zika isolate compared to the other two viruses. The analysis revealed that more than 950 different genes underwent alternative splicing when infected with the modern Zika isolate, compared to just 375 when infected with the original Zika and just over 90 when infected with dengue virus. They also explored the biological processes associated with the genes that were alternatively spliced. One thing that began to stand out was that the most modern Zika virus used in this study resulted in a lot more changes in alternative splicing itself. In summary, this research sheds light on the intricate world of alternative splicing during Zika virus infections and highlights how different strains of the virus can significantly impact the splicing of genes. All of the research presented during this video is part of a larger project aimed at understanding how the virus itself interacts with different cellular processes and how that may influence or lead to Guillain-Barré syndrome, microcephaly, or some of the other developmental disease manifestations associated with Zika virus. In an upcoming video, I'll share with you a different but equally intricate process by which Zika virus interacts with host cellular machinery and disrupts normal processes. If you enjoyed this video at all, learned something new, or found something interesting about this video, consider giving it a like. Stay tuned for more medicine and infectious diseases videos, as well as flight reviews, plane spotting, and everything else aviation related. As always, thanks for watching. See ya.